Hi, my name is Victoria Gustafson. I'm a student at the University of North Carolina at Chapel Hill, and today I'm going to be giving you a simple introduction to the research technique or assay immunofluorescence. I will be applying this technique to research currently being conducted in the Sikelski lab at UNC, which focuses on molecular genetics. Let's jump in. I'm going to start by taking a step back. You might be wondering, what is fluorescence? Well, fluorescence is a type of luminescence caused by a specific emission of light. Broadly speaking, it's used in biology research to identify specific structures in tissues and cells. You may have seen fluorescence in some deep sea creatures, such as jellyfish, as seen right over here. When jellyfish are exposed to a specific light source, ultraviolet light, they have this sort of glow to them. If you end up taking an analytical chemistry course or a physical chemistry course in college, you'll learn what happens at the atomic level that causes fluorescence, the specific mechanism. But that's beyond the scope of this introduction. So for now, understand that fluorescence is a specific molecule emitting light. So in the Sikelski lab, fluorescence is a way biologists can visualize specific parts or molecules in a tissue sample or cell. And it's applied to cells through the method I'll be talking about today, immunofluorescence. While the word immunofluorescence may seem daunting at first, I want to remind you that it's actually pretty cool. Here is the result of an immunofluorescent assay in a model organism called C. elegans. And these biologists wanted to visualize a protein found in the worm's nervous system. So the nervous system is what's glowing in this picture. Now, let's learn how biologists are able to do this. So biologists use immunofluorescence if they want to visualize the expression and location of a specific protein in a cell or organism. Immuno refers to the antibodies used in this technique, and fluorescence refers to the emission of light. So specific antibodies selected by the researcher bind to a protein of interest, the protein that the biologist wants to see. And fluorescent dyes are coupled to these immune complexes to visualize the protein of interest. So there's direct immunofluorescence, in which a antibody connects to an antigen present on the target protein, and connected to that antibody is a little fluorophore, which is just the molecule that is fluorescent. That's direct immunofluorescence. And then indirect immunofluorescence just has two parts to it instead of the one. So one antibody, the primary antibody, binds to the antigen on the target protein, while a secondary antibody binds to the primary antibody, and it's the molecule that holds the fluorophore. To accurately see fluorescence, biologists use a microscope and can take pictures of their observations using a special camera that captures a microscope's view. So in the Sikelski lab, the specific microscope that they use is a Zeus LSM 710 contrifocal laser scanning microscope. I know, sounds fancy, but this is an image of it. And it visualizes fluorescence, and the pictures are collected, as you can kind of see on this computer, using Z-Sense software. And it looks just like a kind of a regular compound microscope connected to a computer. So here are the results of an immunofluorescent assay conducted by biologists in the Sikelski lab. You may notice the different colors, signifying different parts of, in this case, the nucleus of the cell. So the picture labeled A shows a normal nucleus of a fruit fly, while the picture labeled B right below it shows the nucleus of that biologists experimented with before capturing the images. So as you can see, there are clear observational differences from these two pictures, and that can help biologists understand how different genetic processes work. Okay, I hope you enjoyed this simple introduction to immunofluorescence. And to finish, I'm leaving you with this super cool capture of a jellyfish from the Monterey Bay Aquarium. Have a wonderful day.